Greetings everyone, this is Brian Cary. I'll be a guest speaker for this video on this week's technique, extracellular recording in vivo. What is extracellular recording and how does it differ from other types you've learned about? You should be acquainted with intracellular recording from the patch recording technique video. Here is an idealized action potential as it would occur if you were recording from a single neuron with patch electrophysiology and current claim. The intracellular potential goes from typically negative, sharply to positive, and quickly back down. You can think of this as the intracellular perspective. If you imagine that instead of measuring the potential or voltage from the inside, you had the electrode outside of the cell, but nearby, this action potential or spike, as it is often called, appears flipped. And to make this make sense, think about the movement of ions. The positive ions that are flowing into the cell are leaving the extracellular space, making it more negative. This produces the characteristic negative spike of extracellular recording. So why record in this way, from outside of the cell? Intracellular patch recording has the advantage of providing highly specific and sensitive measurements. You can record very small synaptic events and even single channel currents, measurements not typically available via other methods. Intracellular recording also allows for a tremendous amount of control over a neuron. For example, a large range of electrical and chemical manipulations. However, as you might imagine, performing intracellular recording, typically with a glass pipette, is extremely delicate, and it can be a relatively cumbersome technique, making it far less viable in the behaving animal. Additionally, extracellular recording makes it possible to perform far larger scale recordings with dozens to even hundreds of channels, and all within the intact brain. There are three general stages of extracellular recording. First, the animal will undergo surgery to implant the electrode. To do this, an experimenter will need to have been trained in proper surgery technique, including use of anesthesia, aseptic technique to reduce chance of infection, and post-operative care to ensure full recovery. After electrode implantation surgery, the animal will be hooked up to a data acquisition rig where the channels will be recorded from during a given behavior or paradigm planned for study. Finally, once recording is finished, typically an enormous amount of data has been collected, and data analysis takes place, which can be further broken up for our purposes into spike detection, feature extraction, and clustering. For this presentation, we will focus on single cell activity, or what is often referred to as single unit analysis. Typically, around half an experimenter's time or more might be spent on just this final step of analysis. I'll discuss each of these stages now more closely. Surgery starts with prepping the animal and administering general anesthesia to induce unconsciousness. An incision is made to part the skin and expose the skull. Next, commonly after making measurements to target a specific brain region, a craniotomy is performed to remove a portion of skull above the brain. Using a stereotex, like the one pictured earlier, an electrode drive is slowly lowered into the brain. After electrodes are lowered to the desired location, wires from the drive are connected to a screw or two in the skull to electrically ground the electrodes during recording. This can reduce electrical noise. Finally, surgery cement is applied to fill in the exposed area to seal it off and firmly hold all the parts in place. Once the animal has recovered from surgery, the animal will be connected to a recording setup. The voltage signal is recorded at a rate of usually 20 to 40 kilohertz, that is 20 to 40,000 samples per second for each channel. electrode sites can number from several to now many hundreds. For this reason, the size of the data collected is very large, within the gigabyte to terabyte range per experiment. The raw analog signal is amplified to improve the resolution of the signal. And then the data is converted to a digital format and stored on a computer nearby, often with other behavioral data like video of the animal. Two popular types of electrodes used in vivo recordings are silicon probes and tetrode drives. Silicon probes are fabricated using automated machines and can be made in a range of shapes and sizes with precise dimensions. They are usually expensive, but are highly standardized. Here is a schematic for a specific probe, and here is an actual picture of what they might look like. Tetrode drives, however, are often hand-assembled and consist of multiple sets of wire bundles, each with 4 electrodes. The tetrodes can be individually positioned by hand after implantation into the animal. The 4 electrode sites help ensure that nearby neurons can be independently identified later in analysis. And here is an example of what a tetrode drive might actually look like. Each channel will record a voltage trace, like one of these shown here. You'll notice that in the raw data, there is a slower change in the voltage that results in this wobbly baseline that goes up and down. In addition, if there is nearby neuron activity, you'll see these very quick spikes. In order to perform single unit analysis, that is to look at the activity of single neurons, it's important to first filter this raw signal. By passing the data through a high-pass filter, we can remove the slower fluctuations in traces and preserve just the quick spikes. Next, you can mark all the times that the filtered signal crosses a voltage threshold, usually defined as a number of standard deviations. This simple process allows us to detect and extract many of the spiking events. Each vertical line marking here is a spike, and as a reminder, if we zoom in, they will generally take on this upside down waveform introduced in the beginning. At this point, I'll introduce a very important detail concerning in vivo recording as described. Because each channel is simply inserted into a brain and not targeted to a specific cell, as is done with patch recording, each channel will often pick up the activity of multiple nearby neurons. For single unit analysis, the desire is to look at the activity of individual cells. But if each channel detects spikes from multiple cells, how can we know which spikes belong to which neurons? You might be able to see that among these detected spikes, some have different heights. Fortunately, the waveform, that is the shape of the spike, depends greatly on the relative position of the electrode to the neuron. This graphic shows the simulated shape of spikes from this neuron with different electrode placements. As you can see, the same spike appears different depending on where exactly the electrode is. This means that while a channel might pick up the activity of multiple cells, each cell should have a unique waveform. The second step attempts to address this complication by extracting different features from every single spike waveform in order to estimate which spikes belong to different cells. Common features to track for each spike include the amplitude of the first downward spike, the width of the spike in seconds, and the time it takes to go from the trough to the positive peak, often occurring after each action potential. These days, often more advanced computational techniques are used to extract additional, less obvious, but useful features in each spike. After collecting features of every spike, one can compare them using 2D or 3D plots, or using different metrics that measure similarity, boiling it down to a number. This plot shows the amplitude of spikes from 4 identifiable neurons on 2 nearby channels on a tetrode, amplitudes recorded on channel 3 on the x-axis versus those on channel 4 on the y. All 4 of these neurons are near the 2 channels, but show different amplitudes and shapes, which can be seen in these plots. Each dot represents a single spike, and they are colored based on which cell they are estimated to belong to. Gray dots represent spikes that cannot be easily classified or are noise. Clustering gets its name from these groupings that occur in the data points. At the end of this data analysis pipeline, you will have recorded the precise timing of spikes from putative single cells. This type of extracellular recording allows you to collect data from a relatively high number of neurons and from the intact brain of an animal as it behaves. One common analysis done using this data with combined behavior is to create a peristimulus time histogram, or PSTH. This can be created by recording the activity of a neuron over many trials of some behavior. Here is an example figure from the paper for this week. In panel A, each black dash represents a single spike, and each row on the y-axis is a round of behavior. Below, in panel B, is this same data represented with a histogram, where the number of spikes are counted in each of these bins. A PSTH can be interpreted as trying to define the average response a neuron has to some behavioral feature, which could have important implications for the neuron's role in that behavior. Recent rapid developments in technology have made massive recordings possible. Here is a raster plot from one animal where again each minuscule dash is a spike, but this time each row is a new neuron. There are over 800 neurons shown here, recorded simultaneously from just a single probe, a feat hard to imagine 10 years ago. Data from extracellular single-unit recordings has proven to be a rich substrate for many far-flung hypotheses about what this neural activity represents in the brain and how it contributes to behavior and cognition. Thanks for listening.